Dr. Matthew Benz, who's going to speak on the genetics part of the DNA process that's going on, that's new and going on now. And then we've got Byron Rogers with Pedigree Consultants, who's going to talk about the future and where it's headed. And then Dr. Lambert's going to speak about how it relates to the overall sales aspect. I think, when we started out measuring with the heart scans, and now we're with the DNA part. And I think after the recent sale, we all are probably a little bit apprehensive as to having something else come in that potentially takes a buyer away from one of your horses. So anyway, we'll get started, and we'll let Dr. Benz kind of talk about the genetics part of it, and then we'll roll through them, and any questions y'all can feel free to ask. Thanks again. If y'all want to come up this way, there's plenty of room up this way if you want to come up this way.
Okay, well, I'll get started. I'm sorry if I've got my back to some of you. I can't avoid it, I don't think, so I hope you'll still be able to hear me. I've got a loud enough voice. Thanks to the organisers for asking us in today to talk about genetics and DNA testing. I gave a similar talk last year at the meeting Byron organised, and so I based this talk around that. But I think there's we're tailoring it specifically to your, what we understand are the sorts of questions you have. So, the basic question, genetics, is it important? And one easy way of relating that is genetics is actually pedigree. That's how I think about genetics. But I actually think about it in probably a slightly different way to you guys do. So here's a traditional pedigree used. I'm sorry again, it's a small screen, but it's Zenyatta, so probably most of you know it backwards anyway. And when you look at a pedigree like that, you're probably thinking of horses in that pedigree. You can remember how they raced, you know what progeny they produced, and you've got a pretty good idea of the animals within there. And then I've talked to people who are arranging matings in England for very big clients, and one guy was saying, I like to see doubling up in the same position, top and bottom, and the same position meant something to him. And this was a really successful advisor. So here we've got Nashwa in the 5th generation, doubled up in the same position. And as a geneticist, that doesn't mean a lot to me. And I'll come to that in a second. I love this Peb cartoon, which is in the Keeneland, up in the bar area. You've got two foals talking to each other. One of them's sort of, I'm out by Northern Downs, here's my pedigree. And the other one's saying, so what? And part of me feels like that. Pedigree is only a very limited part. You all know horses with fantastic pedigrees who were totally useless. You also know horses with poor pedigrees who were absolute stars. It happens. When I look at that pedigree, the same thing of Zenyatta, what I'm seeing are blocks of DNA and DNA behaving in a particular way. Some of it's predictable, most of it isn't predictable, just by looking at the names on a page. It is predictable it's totally predictable using the modern DNA tools we've got. So here's a 3-generation pedigree, and I can absolutely follow blocks of chromosomes through pedigrees. And that's the way I start to think about the business we're in. If I look at that traditional pedigree, I can only tell you 2 things. I can follow the top male line and say there's a Y chromosome, passes street cry, Machiavelli, Mr. Prospector, raise a native dancer. The Y chromosome's a tiny piece, it's the smallest chromosome, and it's got about 30 genes on it. It's not likely to be involved in performance. So whilst we make a big fuss about the 3 founding stallions in 1700, 1720, there's probably not a lot of variation amongst those in practical terms of what came down those generations. The one thing it does do is specifies maleness. Single gene on the Y chromosome is a male. The second thing I know about that pedigree from a DNA level is that the mitochondrial DNA follows the tail female line. And if you trace back, you get to whatever family, I can't remember what family Zenyatta is, but some of you might know. Back to one founding mare again in the 1700s. Mitochondrial DNA is a bit more interesting. Again, very small piece. It's got about 29 genes on it, some of which are involved in energy metabolism. There is variation in some of those genes, and some people think they're important to ability. If they are important, we think it's probably a fairly small amount. Mitochondria are really important. They're the energy factors in the cell, but most of the genes for mitochondria are actually on the genome. They're not on mitochondrial DNA. All the rest of the mix and matching in that pedigree, you can't predict. You know that, Zenyatta got half her DNA from street, one copy of each chromosome from street cry and one from Vertigio. As soon as you go back into that grandparental level, you can't say what's come from where. And again, how many times do we read pedigree reports which say this horse got the speed from this individual in the 5th generation, or from doubling up in the 5th generation? You have no idea. You just don't know. But again, to stress, with DNA markers, we can follow it back. And we've got 5 generations of animals, and we can actually follow the DNA through those 5 generations and see what is being passed on. So, I said genetics is pedigree. Is it important to performance? What's the influence on the ability of an individual horse? Well, the scientific studies done in Ireland in about the 80s said that genetics looks like it's somewhere between 1/3 and 1/2 of a horse's ability, and the rest is environment. And environment starts as soon as you've got conception. If you've got a really old mare, you might get a not very good foal out. If you've got a maiden mare, you might get slightly smaller foals because placentation doesn't seem to work quite as well. So that inter-uterine environment is important, but then all the way through. Training, breaking, so many factors will affect the ability and the eventual outcome of that horse. But genetics is something which is solid and we can work with. We know it's there, it's there. It's something we can analyse with some exactitude. So the way I think again about this is, here you are, you've got this animal, bundle of genes, a particular combination. And there are literally millions of different combinations possible. In fact, when you mate any sire with any dam, you've got tens, hundreds of thousands of possible outcomes. And just because it didn't work the first time, doesn't mean it wouldn't work the second time. And that bundle of genes is, again, then wrapped up in this environment to give you the output. And that extent of genetics is called heritability. Next slide, I just tried to emphasise that. I can stand up the front of this one. So here I've got 3 different horses, and these are their rankings. This is how good they are, say. And this is the best one, next best, that's the worst. And it's a mixture of genetic contributions and environmental contributions. And if there were 6 genes, say, affecting performance, you can see that we add up the scores for each of these genes. This one's plus 16, this is plus 5, this is minus 10. And you score these up. This is in a lousy environment, this is in a great environment. And so, just looking at these, you can't say how good the genetics is. If you think about how we select breeding animals, we select stallions, we're basing it on this sort of number here. And what is it? 1 in 50 stallions really makes it as a really big thing. I mean, 1 in 10 probably is commercially successful. But to make a real effect on the population, on the breed, there's not many of these we're picking out. If we could pick out individuals who have good genetics, would improve the outcomes. And that's what I meant to say as well, with that pedigree slide. DNA, it sounds like a mystery, although we're exposed to it in the public eye all the time now, medical things. You're already pulling hairs for parentage analysis. Why do you do that? Well, because you're going to get 1 copy of chromosome from the dam, 1 from the sire. If the foal doesn't have a combination which fits those 2, you know it didn't come from that sire or dam. And I used to run the parentage verification lab in England for a couple of years. I was mainly a researcher, but at one point it became my responsibility to run the Weatherby's lab when they moved from when they were moving from blood to DNA. They had quite a lot of problems, and I came in and made sure that the lab worked well for the DNA. So those sorts of markers that are used in parentage are the same sorts of things, Genetic marker is just something that varies between individuals and lets us identify particular bits of chromosomes. Now, we're interested in performance, and the traditional way is, how do we identify a gene involved in any trait? Well, one obvious way is to use comparative genetics. Do we know about this gene in another species? And I've got an irreverent slide, which I hope none of you object to too much, which says we can take information from the human genome about genes and apply it to the horse. And we know quite a lot about the genes in humans. The second way was to use what was called traditional genetic linkage, and that we needed family material for that. And it's actually quite hard to get large amounts of family material from 2-3 generation pedigrees. They're going to be half-sibling families. But we did this for some of the gene mapping we did originally. The first gene we mapped with linkage in the horse was grey coat colour, and where there was no previous knowledge. And again, I think grey is an interesting gene to deal with, because it came into the breed supposedly through one horse in 1720, an individual called Crab. And so every grey horse you see today, you can trace its pedigree back and you'll get back to Crab. Not only did the grey gene come in once, all the genes you're dealing with were captured, and the thoroughbred's been a closed population for, purportedly for nearly 300 years. I mean, in reality, probably for the past 80, 90 years, there hasn't been outside genes come in. So all the material we're working with is just a shuffle of material which was captured all that time ago. Many of these genes we're dealing with today will have been in the breed 200 years. As has the grey. We use the half-sibling family, so the stallion in the middle, lots of mares, lots of foals. We're able to map the grey gene to a place on what's called chromosome 25. That's the old way of doing it. It was time-consuming. It took a lot of time getting the right family material together. The modern way of identifying genes involved in traits is called association mapping, and it doesn't need related samples. And this is what we've done, and we'll just go back. Essentially, an association study, you can take any two divergent groups. If we're looking at performance, we can take a group of good horses, grade 1 winners, and we take under bias 60s as our poor group. And then we look at the markers in those two groups. Can we find any which are solidly statistically different distributions in those groups? And it is as easy as that. It's just compare those two groups. You'll see some of the groups we compared later. Most of this work became possible. I mean, things move very rapidly in genetics, and the technology is moving quickly. And this is it's actually a thoroughbred mare called Twilight, who was an experimental horse at Cornell University. And this is a horse who was used for sequencing the horse genome, which was completed in 2009. It only took about 6 months for this group in Boston to sequence the genome. And the whole sequence has been made publicly available. And for successful genetics projects, you need 5 components. You need a good set of samples, a good toolkit. You need data quality, you need a lot of data, and you need a lot of people knowing what they're doing. And the group, so working with David and Deborah, we've got about 4,500 samples, and we've actually had whole genome scans done on about 1,000. There's more than 200 grade 1 winners in that sample set. We've got 18 individuals who won US Triple Crown races. We've got some of the most important stallions of the past 50 years. We've got a really incredible sample set. The markers, so because we had the horse genome sequence, a commercial company was able to make these little chips, these little arrays on the left. And there's 12 panels on there. We can look at 12 samples of 12 different horses overnight and get 60,000 genetic markers for each one. You don't know how much that is, but it's an incredible amount of data. When I was doing mapping to map that grey gene, we used 300 markers per horse. So we've gone from 300 to nearly 50,000.